Agrochemical exposure can have major impacts on all members of a honeybee colony, including the most important member, queens. But until recently, few tools have been available to study these effects. This technique allows researchers to expose queens to agrochemicals under controlled conditions that simulate conditions they might experience in the field. It's quite easy to disturb honeybee queen egg-laying behaviors, so it's important to take care when handling workers and queens before and during the experiment. Demonstrating these procedures will be Amy Floyd and Eliza Litzy, laboratory technicians at the Invasive Species and Pollinator Health Research Unit, Honeybee Research Lab.
To begin, assemble queen monitoring cages, or QMCs, by inserting the door panels. Then insert the queen excluder in the feeding chamber door over the feeding chamber to keep the queen from entering and contacting the treated diet. Insert a single ELP. Do not add feeder tubes until after the workers have been added to the cage, and temporarily cover the 4 feeder holes with laboratory-grade tape. After collecting and incubating the wax comb frames containing the capped worker brood from honeybee colonies, brush the closed bees off into an open container lined with an insect barrier paint to prevent the bees from crawling out. Add at least 50 bees by weight to the egg-laying chamber of each QMC. To ensure that a diverse genetic pool is introduced, obtain similar number of worker bees from at least 3 colonies and allow them to mix prior to adding bees to the QMCs. Add the feeders containing sucrose solution, water, and pollen supplement. Use queens purchased from a commercial breeder within 48 hours of receipt. While the queen